going to be talking about Mendelian genetics in this lecture. So when we're talking about Mendelian genetics, we're actually talking about the story of Gregor Mendel. Mendel was born in 1822 to a German-speaking family in the Silesian part of the Austrian Empire. He was a monk, and if you read other sources, they'll say that he was a friar. It doesn't make a difference what his religious duties were or his beliefs or obligations. But he ended up gaining fame and notoriety after he passed away, after he died. And he was recognized as being the founder of the modern science of genetics. So Mendel, he's responsible for the laws that govern the inheritance of traits. And he studied the inheritance of traits by using pea plants. And he developed the laws of inheritance. And between 1856 and 1863, he ended up testing close to 30,000 pea plants. Depending on the sources you read, it could be, some will say 22,000, some will say 26,000, some will say 28,000. Doesn't really make a difference. He used an awful lot of pea plants to be able to discover what he did. So he determined that since peas produce sexually, there must be two characters, okay, that influence each trait. So again, today we call these, what he termed characters, we call them alleles. So again, we get these alleles from egg and sperm, right? So one from each parent. So Mendel stated that physical traits, they're inherited as particles. Okay? And again, what Mendel didn't know was that these particles that he was talking about were actually chromosomes and DNA. So in studying genetics, we need to be familiar with the language of genetics, okay? And that comes from learning the genetic terminology. So the first term that we come to are traits. Traits essentially is any characteristic that could be passed from one parent to the offspring. Heredity is the passing of traits from a parent to an offspring. And genetics is the study of heredity. So if we look over here, this is essentially what Mendel did, right? So what is this? This is a flower, okay? And this is the female part of the flower, and these are the male parts, okay? Both the filaments and the anther consist of this part is the stamen that we call. So essentially, again, the boy parts, we're going to have sperm, and the female part, we're going to have the egg. So what Mendel ended up doing was he used a paintbrush that you see over here, and he used this paintbrush to pick up some of the sperm. And then he moved it over to a different plant, and he impregnated that flower, okay? He pollinated it, he transferred it. Okay, so he transferred the pollen, which carried the sperm, to the female, okay? And this is how he ended up doing his experiments. In terms of the different type of crosses that we have, so we have monohybrid crosses. And in monohybrid crosses, what we're talking about are a cross that involves a single trait. So what are we looking at? Perhaps we're looking at flower color, okay? So flower color, if we're looking at flower color, that's a monohybrid cross. Or are we looking at the length of the flower? Is it tall or is it short? Okay, so these would be monohybrid crosses. Now in a dihybrid cross, we're talking about a cross that's involving two traits. So now we could be talking about the height of the flower as well as the color of the flower. So two traits that we look at in a dihybrid cross. So one of the ways that we kind of are able to predict this is by something that's called a Punnett Square. In a Punnett Square, essentially what we do is on one side, we ended up putting the alleles for the one parent, and on the other side, the alleles for another parent. And then we kind of cross them out. So if you look over here, imagine this would be the allele for the mother, and this would be that for the father. So father has XY, or I'm sorry, big Y, little y, and then there's a big Y, little y. So again, these are both heterozygous. And we'll talk about what this is as we move through the lecture. So essentially, what do we get? We end up getting with two big Y, and then a big Y, little y, a big Y, little y, and two little y's. So this will end up telling us what the traits in the offspring will be when we see them. So what are alleles? So an allele is we can look at this as being two forms of a gene, okay? And these two forms of the gene could either be dominant or they could be recessive. Now, dominant, this is the stronger of the two of the genes, okay? So dominant is the stronger of the two genes, and it's always represented by a capital letter, okay? And this is R in this case, or it could be another letter. It could be an S. It could be a T, it could be a big X, it could be any letter that you want, any shape, whatever you decide, any number. But again, we use letters. Now, for recessive is a gene that shows up less often in the cross. Okay, and it is represented by a lowercase letter. So instead of a big R, it could be a little r, a little s, a little t, a little x. Okay? So lowercase letters represent recessive genes. And remember, recessive genes, they're not as strong. They don't show up that much. Okay? Dominant genes, they're very strong genes. They show up a lot. So we're going to be looking into this, examples of both of these also. In terms, when we use this term genotype, we're talking about the gene combination for that trait. Okay, so what is a gene combination? So is it, remember, genes, one from mom and one from dad. So are they both the same, or are they different? Or how different are they? So we describe each one of these. So this would be, since this is a dominant and they're both the same, we say this is a homozygous dominant. And because this is different, we have one dominant and one recessive, we say this is a homozygous dominant. And because both these are recessive, we say this is a homozygous recessive trait, right? A recessive trait. Now, phenotype, this is what ends up showing what are the physical features that result from this genotype. That is what phenotype is. All right? So, in other words, is it red? Is it white? Do we see pink colors that come from this genotype that we have? Okay? So, phenotype essentially is what? What do we see with our eyes? With our naked eyes, what are we able to observe? This is what genes produce what we see. Okay? That is the phenotype. So when it comes to the genotype of alleles, all right, we're going to be talking about flower color in this example. So if we see a capital R, we're talking about red flower. Lowercase r would be yellow flower. So little r, again, this is yellow flower. Big R is red flower. Now, all genes, they occur in pairs. Remember, one from dad, one from mom. So two alleles, they affect one characteristic. Right? So what are some of the possible combinations that we get in terms of genotype for this flower color? Well, we can have big R, right? And this is homozygous, right? Homo means same, okay? Same zygote, they're the same alleles. Homozygous, this is a dominant, homozygous not dominant. This is a dominant R, and then we have a recessive R. But remember what we said. The dominant allele will mask, it covers up the little one. So we're still going to see a red color, okay? Because this little R, this recessive white, it's weak. This big R is much stronger. It shows over the white. So we still end up seeing red. Now, when we have two little R's, then we get a yellow. I'm sorry, so we're not talking about white, we're talking about yellow. So two little R's, when we have these two little R's, then we get a yellow. That's phenotype. So this two little R's, because they're the same, again, we say it's homozygous. In this case, because it's a recessive trait, homozygous recessive. This is homozygous dominant. And this is heterozygous. They're opposite, right? One big R, one little r. One red, one yellow. So heterozygous, heterozygous dominant. Sorry, yeah, heterozygous dominant. This would be. So, again, we're explaining this. For a homozygous genotype, again, this is when the gene combination involves two dominant or two recessive genes. Okay, so again, it could be two big R's or two little r's. And we also call this a pure genotype. Okay, a pure genotype is also known as a homozygous genotype. Now, when I come to a heterozygous genotype, now we're talking about a gene combination of one dominant and one recessive allele. So you got big R, little r. And again, this is also known as a hybrid genotype. Okay. So why did Mendel use peas? I don't know, peas were cheap. Peas are still relatively cheap. But all jokes aside, the garden pea is an ideal organism to study because it grows so fast. It grows vigorously. It's able to self-fertilize. Right? In addition to that, it can easily cross-fertilize as well. Insects, a bee, wind, they can all cross-fertilize these plants relatively easy. And also, you can produce a large number of offspring with every generation. So these are all the advantages, and again, they were very easily available. So this is why Mendel chose to use this plant. They're easy to grow. So when you look at this, all right, let's take a closer look at this. So the ovary, it contains the eggs that are found inside the flower. Okay, so the ovary contains the egg that's found within this flower. Pollen, it contains the sperm that's produced by the stamen. Okay, so here's the stamen. Okay, and pollen will contain the sperm that's produced by this stamen. Okay, pollen can carry the sperm to the egg. How? Perhaps a bee sits over here and then comes and sits over here. Perhaps wind blows it. Perhaps another animal's walking by and it passes over. So any number of ways this pollen could be transmitted. Again, this is what self-fertilization is. It can self-fertilize. Or again, wind can, or an insect can take this and a bee will come sit over here. It will get the sperm on it. It will get the pollen on it, and then it goes to a different flower, and then again it cross-pollinates it. So this is what cross-fertilization essentially is. Okay, this could be done by wind, or it could be done by some animal that's moving through the garden, or again a bee or any number of other insects are able to cross-fertilize. So for pea plants, there's 8 traits that we can look at. Okay, we can look at the seed shape, and that's, the seed shape could either be round or it could be wrinkled. And again, round, this is the dominant trait, and this is the recessive trait. The color of the seed, the dominant is yellow, recessive is green. The shape of the pod, is it smooth or is it wrinkled? Okay, dominant is the smooth, wrinkled is the recessive. The color of the pod also, when we're talking at green is the dominant and yellow is the recessive. The seed coat color next is gray is the dominant and white is the recessive. The flower position, is it axial, which is dominant, or is it terminal, which is the recessive? What about the height of the plant? Tall is the dominant and short is the recessive. And as far as color of the flower goes, purple is the dominant and white is the recessive. So when you look over here, again, this just kind of goes over what we looked at before. So here we're looking at the shape of the seed. Are we looking at it smooth or wrinkled? What about the color of the seed? Is it yellow or green? And the shape of the pod could be either inflated or it could be wrinkled. The pod colors could also be either green or yellow. Unfortunately, it's not in this picture over here. But we can see the color of the flower. It's either purple or white. And also we can see the flower position. So the flower position, it could be either axial or it could be terminal, as we see over here. And next we look at the length of the stem length. It's either a tall or it could be a dwarf or a short. Okay. We have more terminology to look at. So now we're looking at terminology in breeding. So the first generation or the parental generation could also be, it's P over here, or also you can look at it can also be said to be P1. Okay, so P1 or P generation, this is the true breeding. This is the parents, the parental generation that's in the breeding experiment. Now the first one, okay, the F1, this is going to be the progeny, okay, or the first generation offspring in a breeding experiment. And this is also what we call the first filial generation, okay, the first filial generation. Now, F2, the second generation, this is the offspring this is the second generation offspring in the breeding experiment. And that's F2. Or also we can say it's the second filial, okay? So now, when you look at the third generation offspring in this breeding, in any given breeding experiment, that would be the F3 or the third filial. Or again, when you look at the fourth generation offspring, that would be F4, and F5 would be the fifth, and on and on and on. Okay, so this is the terminology that we use. F1, F2, F3, F4, F5. The first filial, second filial, third filial, fourth filial. The P, the parental, P1, the parental generation. So, following the generations, let's move along. So we have a cross between two pure plants. We have this plant here, which is purple, and then we have another one which is white. Okay, so this would be represented by these two dominant genes, and the white would be by the little p's. So what do we end up getting? Well, we end up getting, let's make a cross over here, huh? How about that? So we can get some practice. So we said one parent, let's just say this is mom, is big P, and then let's say dad over here is going to be little p, little p, little p, little p. So what do we get? We have a big P and a little p, a big P and a little p, another big P and a little p, big P and a little p. So essentially we have all heterozygous dominant offspring. And again, what do we get? This is this F1 generation. Now, if this F1 pollinates, reproduce asexually, what's going to end up happening? So let's make up a square for that now. So here we go. Now remember what we said. It's going to self-fertilize, right? And what do we have? What is the genotype? Big P, little p. You can see it over here also. Big P, little p. Big P, little p. Okay, so now let's see. Little p, all right. So you got one big P, one big P. We got one big P, one little p. We got another one big P, another one little p. And then we got two little p's over here. So in other words, as far as the phenotype goes, what we're going to see, since there's two little p's, there's only one of them with two little p's, so this is going to be this one over here. The rest of them, yeah, we have one where it's big P, and then we have two that are big P, little p. So let's actually write this out. We have one big P, which is this one here. Then there's these two over here, which are big P, little p. And then we have one little p. There's one over here. And we said there's two over here and there's one over here. So the genotype ratio is 1 to 2 to 1. This is the genotype ratio. And the phenotype ratio is what? 1, 2, 3. Phenotype ratio is 3 to 1. So again, this is more or less the same thing that we kind of looked at on the previous slide. And what they're showing you over here is that we're looking at now, instead of the color, we're looking at if the seed is smooth. Two smooth seeds over here, right? So again, remember, this is a gene, right? Over here, this part is a gene here, this part is a gene over here. This is part of the chromosome. So one is from dad, one is from mom, okay? Or does it make a difference which one? This could be mom, this could be dad. So one of the genes on one of the chromosomes, it's a big S, and the other one, it's a little s that we have, okay? So little s here and a big S there. And then for the other parent, it's the same thing, a big S and a little s. So this is what we call this heterozygous dominant, okay? So when you have two parents that are heterozygous dominant, what do the offspring look like, okay? What is the F? So again, this is the F1, so F1 that we're looking at over here. So what is the F2? What is the F2, what's the second generation going to be? So here, again, this is mom over here. So we have big S gives us one big S, little s, one big S, little s, big S. Okay, that's another smooth over here. And then finally we have little s, which gives us one wrinkled gene, or wrinkled pod over here. Okay, so essentially, when you look at the phenotype over here, it's a 1, 2, 3 to 1 ratio also. So if you look over here, phenotype, 3/4 are smooth, 1/4 is wrinkled. And when you look at the genotype, it's at 1 to 2 to 1 again, right? A quarter is big S, it's homozygous dominant. Half is going to be heterozygous dominant, and then the remaining quarter is going to be homozygous recessive. So again, this is that same, actually it's the same thing. So they're talking about, so this is smooth seed. All right, so now they're talking about it's the same thing. The seed shape, is it round or is it wrinkled? Okay, so instead of using S as they did over here, they're using R over here. So what do we see over here? We have big R, this is from, let's just say this is dad, and let's say this is mom. And mom has two little r's, right? Mom is going to be wrinkled and dad is round. So when you cross this, what do we get? So look, big R, little r gives us RR. You take this big R, this R, RR, this one and this one, RR, this one and this one, RR. So what do we see? The genotype are all the same. The genotype is all heterozygous dominant. Okay, so what is the phenotype? What are they all going to look like? They're all going to look the same. They're all going to be round phenotype. Now, what's the ratio? 1, 2, 3, 4. They're all alike. So again, 100%. What about the phenotype ratio? They all look alike. So 100% same. Okay, so remember, this is what we see in the P1 monohybrid cross. Now, so again, we looked at these terms over here, homozygous dominant and homozygous recessive. This is what we crossed. Homozygous recessive over here. The mother is homozygous recessive with the homozygous dominant father. So the offspring are all heterozygous. They're all hybrids, right? So this is what we call heterozygous dominant or hybrids. So all these four are what is all, not only, we also say this is the first generation or the first flail. And genotype and phenotype ratios, they're all the same, right? Because why? They're all RR, and again, what they produce, their genotype and phenotype are the same. So now let's go to the F1 monohybrid cross. So again, let's do RR and RR. So let's take these guys and pollinate them. Let's cross them and see what we get. So here's dad, and let's say this is mom. So again, let's do the math. Well, not the math. Let's do the cross over here. The test cross. RR, big R, big R. Big R, little r, big R, little r. Little r, big R, we get this. And little r, we get this over here. So what's the ratio that we have now? let's look at the genotypes first. Okay, so first of all, okay, the genotype that we have, big R, we have big R, or we have little r. Phenotypes are going to be what? Round and wrinkled, right? They can either be round or they could be wrinkled. Now let's look at the ratios. So we have one big R, that's our 1. We have two big R, little r's, that's the 2. And then we have one little r. This is this one. So the genotype ratio is 1 to 2 to 1. Now in the phenotype ratio, remember these three, they're all dominant. So we have 3 over here, 1, 2, 3. And we only have one that's wrinkled. This is the one, a 3 to 1 ratio. So again, to review, what do we see? 25% we see homozygous dominant, 50% heterozygous dominant, 25% homozygous recessive. What was this, the offspring called? This is the F2 generation that we have. The genotype ratio is 1 to 2 to 1, and the phenotype ratio was 3 to 1. So, when it comes to the test cross, Mendel crossed a pure and a hybrid from his F2. So this is known as this F2 or test cross, and we end up getting 2 possible test crosses. So let's look at that now. So looking at the F2 monohybrid cross now, we have this one parent, which is big R, little r, and then in other words, we have a heterozygous dominant parent, and then we have a homozygous recessive parent. So what do we see over here? What do we have in this F2 cross? So again, we have one big R, little r, we have two little r's over here, and then over here we get one big R, one little r. Then over here again we have two little r's. So let's look at our genotype ratio. We have 2 big R, little r. In other words, we have 2 heterozygous dominant, and then we have 2 homozygous recessive. So again, it's 2 to 2, reduces to 1 to 1. What does this produce? This produces wrinkled. So we have 2 wrinkled, and then we have these guys over here that produce round. It's 2 round. So again, we have a 1 to 1 ratio. 50% are wrinkled and 50% are round. So to go over this, the inheritable factors or genes are responsible for all inheritable characteristics. Why does it say like this? Inheritable. Okay, it should be inheritable. All right. So the phenotype, what we see is based on the alleles that are present, right? Which is what we refer to as the genotype. So each trait is based on the 2 genes, one from mom and one from dad. Now, when we have true breeding individuals, in other words, we're talking about homozygous. Okay, both alleles are the same. Okay, this is homozygous. And if they are hybrids, then they have different alleles. Both alleles are not the same. Okay, so those are called hybrid. So homozygous versus hybrid. So you should have a good understanding of this. We went over all this in the previous slides. That was the terminology. Now, when it comes to the law of dominance, so in a cross of parents that are pure for contrasting traits, only one form of the trait ends up appearing in the next generation. Okay? Only one form of the trait will appear. The offspring, they're all going to be heterozygous, and they only express the dominant trait. So if you look over here, again, what are we saying? For pure contrasting traits, only one form of that trait will appear in the next. Okay, so now we have this one and this one. What do we all get? They all produce one trait, which is what? Round seed. You don't get both. We only get one. One dominates over the other. This is what they're saying. Law of dominance. You can't get both. So if you look over here again, we have two different type of genes over here, a dominant and a recessive, but law of dominance says that only one trait will be displayed, right? So this is yellow or either green. You don't get half yellow, half green. That doesn't happen. Well, actually, it does kind of happen, but again, we're not going to talk about that now. It's extremely rare. You're not going to get somebody with one blue eye and one green eye. Perhaps in a very, 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 very rare situation that may appear, but again, otherwise, no, it's not normal. Okay, so again, law of dominance, we only see one trait that appears. Moving along, law of segregation. So what does law of segregation say? So gametes that are formed during meiosis, they end up separating alleles so that each gamete contains only one gene for each trait. And at fertilization, the alleles, they combine to form new combinations. And Mendel, he tested this hypothesis by predicting the outcome of the crosses he never did before. When we look at the law of independent assortment, what we're saying is that two traits will be inherited independently of one another, provided that their genes are located on non-homologous chromosomes. So when gametes form, the alleles for the different traits, they segregate independently of one another. Okay, and again, we can understand this by looking at a dihybrid cross, and which is what we're going to be looking at now. So in a dihybrid cross, again, now we're looking at two things, not one. Remember, di means two. Di means two, so dihybrid cross. Now what are we looking at? Seed shape and seed color. So it could be either round or wrinkled or yellow or green. So this is one parent here, and here's another parent over here. So what is this? This is going to be a heterozygous dominant. This is also heterozygous dominant, two heterozygous dominant parents. What are the possibilities from each parent? So look, big R, little r. It could be, you can have one big R, one big Y, or it could be one big R and one little y, or it could be, again, a little r with a big Y, or it could be a little r with a little y, right? And the same for this parent here also. These are all the possible combinations of gametes that we have. So now let's just take this and cross it. Cross them all. Okay, that's all that's being done over here. So I'm not going to go through all these, but instead, let's look at the outcomes instead. Okay, so remember what we said. Big R is round, little r is wrinkled. Big Y is yellow, and little y is green. So we got two big R's and two big Y's. What does this mean? This means round and yellow. Okay, so let's do this. One. This is also going to be round and yellow. Two. This is also going to be round and yellow. This is also going to be round and yellow. Now let's go over here. Round and yellow. This is 5, sorry. This is round. Is this round and yellow? No, this is what? Round and green, right? Two little y's. Let me just put this on so it's easier to look at. Round, actually, it doesn't make a difference. All right, so here's another one. Big R, little y. This is going to be our number 6. This is not round and yellow. This is round and yellow, so that's 7. And this is also round and yellow. And we have one more round and yellow over here, 9. Okay, so now 9, this is our 9, round and yellow. Now let's look for the round and green. So here, this is round and green, 1. And round and green, 2. And round and green, 3. Okay, so we found our 3 round and green. Now we're going to look for wrinkled and yellow. So here's a wrinkled and yellow over here, 1. Here's another wrinkled and yellow, 2. And here's another wrinkled and yellow, 3. Here's our 3. And what do we have? Wrinkled and green. Wrinkled and green, 1. So what's the phenotypic ratio? 9:3 to 3:1. Okay. All right. So now we're looking at this test cross, a mating between an individual of an unknown genotype and a homozygous recessive individual. All right, fine. So this is just a problem that they're giving. So what are we looking at here? Eye color and here. So for eye color, it could be either brown eyes, which are 2 big B's, or you're looking at, this is a homozygous dominant, right? Homozygous dominant. This is a heterozygous dominant, big B, little b, and then we have a homozygous recessive, which is 2 little b's, 4 blue eyes. Then when it comes to curly hair, to here, curly hair is dominant. So 2 big C's is curly hair, a big C and a little c is going to be curly hair as well. And then if you want straight hair, then that's 2 little c's. Okay, 2 little c's. So let's make an example over here, huh? So we have one parent here that's BC, BBC. Here we have one parent that's blue eye and little c, that's straight hair, huh? Blue eyes and straight hair. Little c, we said was straight hair. Okay, so now let's just guess something. Let's just make it up. It doesn't make a difference what. Oh, let's just make this, let's say, fine, let's do little c. Okay, so what do we get? We'll make this little c. What's the cross look like? So you got big B. Let's do this one first. Little b, big C, little c. Now let's do with this guy over here. These two. Little b, little c, little c. So what do we have? We have one that's blue eye, straight hair, and one we have blue eye, curly hair. Okay, that's it. Now when it comes to incomplete dominance, so incomplete dominance, we can also refer to this as a blending inheritance. So sometimes an allele is only partly dominant over another. Okay, so let's see. So in a heterozygote, the dominant allele is only partially expressed. So the phenotype is between the 2 homozygous forms. So this example that they gave over here is this of this snapdragon flower. So we have red, which is by this 2 dominant genes, or it's white. So what happens when you cross these 2? What ends up happening in incomplete dominance is you don't really see the phenotype. You see the phenotype is somewhere in between the dominant and the recessive. Okay, so it's between the 2 homozygous forms. So it's the offspring, they're not going to be red and they're not going to be white. They'll be pink. We get a different color. Okay, so if you do the cross over here, big R, little r, big R, little r, big R, little r, big R, little r. Okay, we end up seeing pink, not red. This is what incomplete dominance is. Now dominance, on the other hand, so 2 alleles, they're expressed. So the multiple alleles. So 2 alleles are expressed in a heterozygote individual. So in dominance, or codominance. So codominance, some traits, they get controlled by 2 different dominant alleles. That's what codominance is. So both alleles are dominant, and there are 2 dominant phenotypes that we have. The heterozygote expresses both of the phenotypes at the same time. All right, so we're going to be looking at the example of blood type with this. So for blood type, Again, what are the blood types? Type A, type B, AB, and type O. So if you're type A blood, then you have the immunoglobulin, and then you have A antigen, right? Immunoglobulin A. Or the gene that could be present would be an A, and doesn't just, this is little I that they have denoted here. For type B blood, you need to have the big I and the big B's, two big B's. Or just one big I with the one B. And then the other one, nothing, it's empty, it's a little I. For type AB, you end up having both A and B. And then for type O, it's not missing this A or B. Okay, it lacks both of these. Okay, so this is what they're talking about codominance is, all right? Now, let's look at the problem. All right, codominance problem. Let's say this is the father, and let's say this is the mother. Okay, doesn't matter, it could be both ways. When you cross this, what do we get? I, big B, and then big I A, big I, B, little I from here, I, B, and I A from here, and then little I from here, and I B from here. Okay, so what do we end up getting? 1, 2 that are type B, and we end up getting 2 that are type AB, right? So 50% AB and 50% B. Okay, so this is what ends up happening when you get a heterozygous, well in this case a heterozygous female with, well, it doesn't make a difference. So when you cross a heterozygous type A and a homozygous type B, these are the results that we end up having. Okay, so this, it would be good if you can try this at home also. If you know what your mom and your dad's genotype are, or if you know what their blood type is, you can kind of pretty much guess, if they're type A, or again, if they're type AB, then you can kind of do the cross and figure out amongst your siblings, if you have siblings, what their probability is of them being, the possibilities. So if you have, for example, if your mom's a type A, and here's a test question for the final. If you have a mother that's type A and a father that's type B, can you have a sibling that's type O? Okay, and if you do, what does that mean? So that's a problem to solve. All right, moving along. Next, we move on to sex-linked traits. So sex-linked traits are genes that are located on sex chromosomes. So remember, in this world, there's boys and there's girls, there's males and there's females. So what determines a male from a female is, whether the chromosomes, of course. So in humans, let's just talk about us. We are said to be male, biologically males. Okay, a biological male is one that has on the 23rd pair of chromosomes that's present, one chromosome is X and the other is Y. Okay, and for a biological female, on the 23rd pair of chromosomes, both of the chromosomes are X. Then you're said to be a biological female, or if you have an X and Y, then you're a biological male. This is the genotype for determining the sex of a human individual. So many of these sex-linked traits, they're carried on the X chromosome. So the perfect example, or one great example, is the eye color of fruit flies. Okay, Drosophila Melanogaster. Okay, and we find that it's linked to the sex chromosome for them. Okay, it's on the X chromosome. You don't find it on the Y chromosome. but it's on the X chromosome. So let's take a closer look at this. So again, these are chromosomes. This is going to be that of the female. Okay, these are both X chromosomes. And you come over here, this is the X chromosome, this is the Y, so this is the male. Female, male. Y chromosome is little, small. And we're saying that on the X chromosome are the alleles for eye color. Okay, so look at this. Mom has alleles on both of her chromosomes, but in the male, only the X chromosome has the color for, or has the allele for eye color. Okay, so let's see how that affects. So here we go. In Drosophila Melanogaster, okay, which is known as the fruit fly, Drosophila Melanogaster. Okay, Drosophila Melanogaster, that's a fruit fly. Don't forget that, make sure you know that. So, eye color in fruit flies. Okay, so what do we have over here? We have red eyes, a red eye male. How do we know this? Because remember, big R is the dominant allele, okay? And this is a male because this is a Y, okay? So this is a red eye male. And then we have a white-eyed female. How do you know this is a white-eyed female? Well, because there's two X's, which means that she's a female. And the little R's are the recessive gene. So female, this is for white, and this is for female. So again, if you look over here, two homozygous dominant is red eye, heterozygous dominant red eye, homozygous is white eye, homozygous recessive is white eye. So now if we have this red-eyed male, which is this one, and the white-eyed female, let's do the cross and see what we get. Okay. Big X, okay, big X, little r. So from here we end up having a big X, big R, and then from up over here we get a big X, little r. Make sure you guys do enough practice of these. Expect them on your final exam. So let's do this over here. Not these, but in general, all the Punnett squares. Make sure you're able to do that. We have a big X and a little r, and then again we're going to take the Y from here. And then let's do this Y, and we'll do this X now. Okay, so big X, little r. So this is the results that we get over here, okay? So what does that give us? Let's see this. Let's look at the next page and see if it's right. All right, here we go. Sex-linked trait solution. So let's see what we have. Over here we have big X, big R, little r, big X, here. Big X, big R, big X, little r, and big X, little r, Y. This matches what we have over here also, right? There we go. Now let's look over here. Big X, big R, big X, little r, big X, little r, Y. Same over here. We've got the same thing. Good. We're matching. So what does that mean? We have, this is our female. Both of our females, in terms of genotype, are the same. Okay? So therefore, their phenotypes will be the same as well. So in other words, we have two females that are red-eyed females. Then when you come to the males, both males are white-eyed. Okay, so we have 50% red-eyed females and 50% white-eyed males. So what cross do you need to have? What do the parents have to be in order to get excuse me, what does a cross have to be in order to get a female that's white-eyed? How can you do that? How can you produce a white-eyed female? Okay, or how do we produce a red-eyed male? So perhaps you can work on this. Maybe you'll see this on your exam. So moving on to multiple alleles. Now, when we're talking about multiple alleles, we're gonna so what do we know so far? So Mendel just studied two alleles in the pea genes, when he was working with the pea plants. However, in real world populations, we often have many multiple alleles of a specific gene, of any given gene, right? So Mendel's work, it suggested that just two alleles existed for each gene. And we know that's not the case, right? So today, that's not always true. And again, usually it's not true. So again, when you look in human populations, humans can, although we can only have two alleles for a given gene, there could still exist in the population multiple alleles, right? And again, these different people in that population, they'll have different pairs of these alleles. So if we look at rabbits over here, right? So let's apply this concept to rabbits. So when you look at this gene over here, this C gene, this comes in 4 different alleles. So we either have this big C, we have the little c, CH, the little CH, or then it's just the little c by itself. So these are the 4 different, the common alleles that this gene comes in. So what are the different phenotypes that it produces? If you look at it, we can end up having a black coated or a black fur rabbit that has this dominant allele, this capital C, big C. Now, the other allele, if it has this little c, CH, that produces this, this is like a gray color, that they call chinchilla color. Now, if you end up getting this little c with just an H, that gives this Himalayan color. And this is like a whitish gray. It's a color pattern. Maybe the ears are, like I said, there's a white body, maybe with dark ears that you can see over here. And then again, the tail's going to be like a gray color also. So there's a Himalayan, there's a variation. Where on the other hand, if it's just the little c's, then in that case, that produces an albino, where it's a completely white, a completely colorless rabbit. Okay? So this is what we end up getting. A pure white coat for an albino. And then Himalayan's kind of like between an albino and a chinchilla with a little bit of, there's some black that you can see also, that may be present. And then in this case, if you have the dominant allele, then you're all completely black. Now, in codominance, we're saying that some traits, they end up getting controlled by 2 different dominant alleles. Okay, this is what codominance is. In other words, both alleles are dominant, and we end up getting 2 dominant phenotypes that end up getting displayed. So again, another way to look at it is, again, codominance, you end up having 2 non-identical alleles of a pair that ends up showing 2 different phenotypes. In other words, one allele cannot mask, it can't hide the expression of the other allele. And one of the examples that we can use to look at this is blood. Okay, so in human blood, you know that we have this ABO, ABO blood group. And what do we see over here? In this case, we have the A antigen that's present. And if you have the A antigen present, then on your red blood cell, then in the plasma, we end up having the anti-B antibodies. And again, what do we call this? This is type A blood. If you have the B antigen on the red blood cells, then you end up having anti-A antibodies in your plasma. Okay? Now, if you have both A and B antigens, then you do not have any plasma in you do not have any antibodies in your plasma. Now, if you have type O blood, okay, this is what this is. This is type O. Type O blood has antibodies for both the type O blood will have both anti-A and anti-B antibodies because type O blood does not have any antigens on its surface. Okay, so the presence or absence of specific antigens types the blood as being either A, B, or O. So how does this work? Well, this is an example of codominance. So this is what we see. If you have this A allele, then that ends up giving you A. Okay? And then if you end up having this B allele, then you end up getting type B blood. Okay? So you end up getting these B antigens on the red blood cell. Sometimes you can have both the A and the B, and that gives you the AB. Other times you don't end up getting any one of these alleles. And when you don't end up getting these alleles, then you have type O blood. Okay? So this is this codominance that's in play. So if you look at this, here's a problem to work with. If you take a male, okay, that's type B blood, and again, type B blood, his genotype, remember, there's two possibilities, right, for type B. Type B could be either this I, big B, and the little I, or it could be just the two I's with big B's. So in this case, they're showing us big B's. So what do we see over here? Essentially, we're being shown that if we take a father that has this genotype, and then you take that with a woman, okay, again, this is going to be the mother that has type A blood, and her genotype is this big I, A, and the little I. Now, what do we end up getting? What's the cross? So again, here's the mother over here, and here's the father over here. So we end up getting 2 children that are both type AB, and then we end up getting 2 children that are both type B. Okay, so this is what this will produce. So this would be good. You know, you can practice this at home. If you know your mom and dad's blood type, right, you might be able to guess their genotype and then kind of figure out, you know, if you have siblings, what the ratio and the proportion of the blood types would be. So this could be something that you may want to try on your own. So sometimes, you know, if you, for example, your mom is type A and your dad is type B, and you have what would that mean? If your brother is type O blood, okay? Here's a question. Here's a good question, and this question may be on one of your exams. If you have a sibling that's, or if you yourself, or for example, there's a family, and in this family, the mother is type A and the father is type B, and there's 3 children or 4 children, and let's just say 2 of the children are type AB, 1 of the children is type B, and then 1 of the children is type O. What does that mean? What does it mean when we have 1 child that's type O? All right, so this is try to answer this question. So to review the different types of dominance, so essentially, if we have the genotype for blue, what are we going to see? We're going to see blue flower, okay, or blue whatever this organism is. If there's the allele for the yellow, again, we're talking about homozygous, then we end up seeing the phenotype being yellow, regardless if it's complete codominance or incomplete dominance across the spectrum. Now, in the case where we have both a blue and a yellow allele, now if this organism is displaying complete dominance, then we're going to see just a blue. Why? Because we said blue is the dominant allele. This is the dominant color. If this organism is displaying behaviors of codominance, then in that case, we're going to see both. We're going to see a little bit of blue as well as a little bit of yellow. Now, in certain cases, in other cases, we end up having incomplete dominance where we have a completely different color. So in this case, the blue and yellow ends up producing a green offspring. Okay, and this would be incomplete dominance. Okay, so incomplete dominance, what do we see? Incomplete dominance, we see a blend, a mixture of the two colors. Okay, so this is green. In codominance, we see both. We see blue as well as yellow. And in complete dominance, only one of these alleles will be prevalent. This is a human karyotype. In other words, these are all, if you were to look inside one of your cells of your body, and we're talking about what is your genetic makeup, this is what you would essentially see. Okay, again, this is when when the cell is dividing. This is when they condense into these chromosomes. You would find a total of 23 pairs of chromosomes. Okay, 23 pairs. So you have the first pair, second, third, fourth, fifth, etc., etc., etc. Now, they're not going to be lined up like this. They're just going to be randomly mixed around within that cell. They have taken and paired them up for the purpose of, again, producing this karyotype. So what we find is this. In humans, we have 22 pairs of autosomes. Okay, these are the cells that make up your body. They determine various things in your body. They have everything about you. 22 pairs of chromosomes do that. We have one pair, okay, and that is the 23rd pair. The 23rd pair are referred to as the sex chromosomes, okay, and primarily they determine the gender, the biological gender of an offspring. So you're either XY, which is a male, as is in this case, you have an X and a Y. This is a biological male. Or it could be two X's, and that would be a biological female. Okay, so again, this is what the karyotype does. The karyotype produces all the chromosomes that an organism has in his or her body. So out of the 23 pair of chromosomes, one of these pairs, again, they contain that sex chromosome. And remember what we said. That is the 23rd pair of the chromosomes. The rest of them, remember what we said, are called the autosomes. Okay, so those non-sex chromosomes are referred to as autosomes. So we have these sex-linked traits that are associated with the genes that we find on the sex chromosomes. In other words, we find on the 23rd pair of chromosomes. Now in humans, these sex chromosomes, as we saw before in the previous photograph, they're either X and they're Y. Now, because the X chromosome is larger, we end up having a lot more X-linked traits being more common than the Y-linked traits. So now when you look over here, in this photograph, this is a big X. Again, if you just look at the sheer size, this is a Y chromosome, and this is an X chromosome. So this X chromosome is much larger than the Y. The Y, it's just very small. It's a tiny little. So one of the things, now remember, this would be a biological male. If this is we're looking at the 23rd pair of chromosome, and you find an X and a Y. Now we're looking at a biological male. If we had two X chromosomes, then we would say that this is a biological female. So the 23rd pair, the sex chromosomes, is that all they do? Do they only store sex information? Well, no, that's not really the case. So besides sex information, the X chromosome, it holds perhaps about 80,000 genes, right? And the Y chromosome, on the other hand, it has about 200 genes that it holds, okay? So that's they don't only determine if the offspring is going to be male or female, but it has a lot of other information as well. There's a lot of genetic information that's present there. Now, traits and genes that are linked to the X chromosome, we call that X-linked, okay? And traits and genes that are linked to the Y chromosome, we call those Y-linked. So the Y-linked traits, they pass through the Y chromosome. So it's only going from father to son, right? It cannot pass to the daughter. Why? Because daughters do not have Y chromosomes. Daughters only have X chromosomes. And we're saying that a Y-linked trait, this trait is only found on a Y chromosome. So females do not have Y chromosomes. Therefore, they cannot have any Y-linked traits. So if this is a disorder we're talking about, or again, whatever condition this could be, or it doesn't need to be a condition, but whatever trait this gene may contain, the sons of that, of the father that's affected, they all end up getting it as well. Okay, so all sons of an affected father are affected by the Y-linked trait. So one of the things that the Y chromosome has is that the Y chromosome has this SRY gene, and this is the maleness gene. Okay, so this is what triggers maleness in males, in boys. Okay. When it comes to X-linked traits, these are genes that are linked to the X chromosome that produce a certain condition or perhaps a disease. Now, these are mostly recessive. So, for example, it could be baldness. We're talking about hemophilia or even color blindness. All right. So, in this case, when you look over here, the disease or the condition is hemophilia. Hemophilia, you can read up about it, but essentially it's a clotting disorder. You bleed too much, and in certain cases, if it's severe, you may end up bleeding to death. So now, if you take a mother, okay, who is homozygous for this condition, okay, so both, it's a recessive allele, so she's homozygous for this allele. Now, what ends up happening? Okay, so this is a homozygous recessive disorder, all right? So she marries her husband who is normal. He doesn't have this. They have children. Okay, so what are the possibilities for the children? Well, they will have 2 girls and they will have 2 boys. So the daughters, they get 1 X from dad, 1 X from mom. Therefore, remember, this is a homozygous recessive. You need to have both alleles present. So they are only carriers. The father is giving a good, and the mother is the one that's giving the allele that has the defect that's present. All right. So the father's good genes ends up masking the bad one. So the female is just a carrier. Okay, they don't display, they don't have any problems. They have the gene, but they don't have any problems. Same thing with this other female over here. The issue ends up being with the boys. All the boys are affected. Why? Because when you look over here, father does not give the X chromosome to the son. The father gives a Y chromosome. So you end up having a Y chromosome from the father, and then you have only 1 X chromosome from mom. And mom has both X chromosomes with this trait. Therefore, both boys end up having the problem. They are both hemophilic. So this is one of the X-linked traits that is a disorder. Okay, this is a hemophilia. Another one that we can look at is eye color in fruit flies. So fruit flies, we're talking about Drosophila Melanogaster. So in Drosophila, the color for the eye colors are found on the sex on their sex chromosomes. Okay, so when you look at this, what are we seeing? The dominant color is red, and that's symbolized by this capital R. Okay, and the recessive gene is going to be the little r, which is for white. So if there's 2 little r's present, then you end up getting white. Okay, and in this case, again, if it's a female, you need both little r's. So now, what happens when you take a white-eyed female and a red-eyed male? You cross them. So in Drosophila, this is what we would expect. If you have both R's, then again, this is dominant, therefore you have red eye. Big R, little r, you're going to have red eye. If you have 2 little r's, it's a white eye. Okay? And of course, XY is male and XX is female. So, again, this is telling you that we have a red-eyed male, and this is telling us that we have a white-eyed female. Okay? So when you do this cross, go ahead, take the time, pause the video, and do this cross to see what you end up getting. Okay? And in the next slide, we're going to go over the results. So this would be a good practice problem for you. So hopefully you guys pause and you ended up trying to solve this on your own, but this is what we end up getting. We have 50% red and 50% white. Okay, so again, big R, little r, remember what we said. This is the dominant allele. Okay, so big R, little r, we still get red. Okay, and this is what we see over here. Big R, little r, big R, little r. Half of the fruit flies, the F1 will be red-eyed color, okay, and female. When it comes to the males, remember, what did we said? The little r is the white allele. Now, males, they only have one X chromosome and the other is Y. Therefore, both males will be white-eyed. So you end up half red and half white, okay? So this is another example of sex-linked trait, is this eye color. Moving on to plyotropy. So some genes affect many different characteristics, not just a single characteristic. So this picture is showing you that, again, one gene could have 1, 2, in this case, maybe 3 features, 3 different features, or it could be 4 or 5 different features. So an example of this, so again, plyotropy, essentially what are we saying? We're saying that one gene could affect multiple features. And an example of this would be something that's called Marfan Syndrome. So in Marfan Syndrome, it ends up, the chromosome 15 is affected, and it's this FBN1 gene that ends up having a mutation. It's a fibrillin gene that's mutated. And what ends up happening is that in these individuals, they're usually very tall in their height. They have thin fingers and toes. There could be problems in their eyes. There could be lens dislocation. They may have myopia. In other words, they may be nearsighted. Aside from that, they could have heart problems as well. They might be hyperextensible for their joints as well. They may have what else? In their mouth, their palate could be. It could have a very high arc, as you can see over here in this photograph that they have over here. So yeah, this is an example of Marfan syndrome. And unfortunately, these people, they don't live very long. They, perhaps they die in their 30s to they're usually in their 30s, their mid 30s, perhaps into their 40s. Moving on to polygenic inheritance. So some characteristics are said to be polygenic. Okay, so poly means many, genic means pertaining to a gene. So pertaining to lots of genes, polygenic. So in other words, some characteristics, they're controlled by many different genes. And in polygenic inheritance, traits often form a phenotypic spectrum rather than just falling into clear-cut categories. So again, we have lots of different skin colors. Look, there's 6 different types of skin colors over here. All right, this is an example of polygenic inheritance. Eye color is another example of polygenic inheritance. Even risk of disease and height, these are all examples of polygenic inheritance. So again, many factors could affect this. These factors, they could be genetic, they could be environmental, or they could be both. So just to go over one more time, because a lot of people end up making mistakes about plyotropy and polygenic inheritance and codominance and incomplete dominance. So again, the major difference between the two is that plyotropy is when one gene affects many characteristics, such as we saw in Marfan Syndrome. And in polygenic inheritance, again, look at the name. Poly means many, genic means gene. Many gene, inheriting many genes, is when one trait is controlled by many genes. Okay, so this is the difference between plyotropy and polygenic inheritance. For the other, codominance and incomplete dominance, remember, in codominance, neither allele is dominant over the other. So both end up getting expressed equally in heterozygotes. Now in incomplete dominance, we end up having an intermediate heterozygote. Okay? In other words, you have a red flower and then you have a white flower. And what do you get between the two? You end up getting, when you cross them, you end up having a pink flower that results. Okay? So this is incomplete dominance. You do not see, both of them are not expressed. You don't see a red that's expressed, nor do you see a white that's expressed. You see something that's something blended and in between, okay? An intermediate that ends up resulting. An intermediate phenotype that results, a pink colored flower. Some genes have alleles that prevent survival when they're either homozygous or heterozygous. And a classic example of an allele that affects survival is this lethal yellow allele. Okay, the lethal yellow allele. So, and again, this is a spontaneous mutation that is found in mice, which makes their coats, again, their fur, yellow. So mice that are homozygous, okay, and homozygous, this is an example right over here, okay, homozygous, they end up, they die. So when you have this homozygous genotype, they die in development. They're never born, okay? So, and then what ends up happening with the rest? The others are carriers. So, you know, regardless if they are a female or a male, they will be carriers. It's not sex linked. So, yeah, this particular allele is dominant. Lethal alleles, they can be dominant or they could be recessive. And they can be expressed in homozygous or heterozygous conditions as well. All right, so again, this is a dominant lethal gene. What this means is, again, only one gene you need, and it ends up, you need, there's only one gene that affects it. It's not multiple genes that ends up affecting it. So if both mom and dad pass this gene along, then unfortunately the offspring will not be viable. As an embryo, it ends up, it does not survive, it terminates. So most real-world characteristics, they're determined by not just the genotype, but also by environmental factors that influence how the genotype is translated into a phenotype. So in this example that you see over here, we have this hydrogena flower. And hydrogena of the same genetic variety, they could vary in their color from blue to pink. And this is dependent upon the pH of the soil that they are in. So again, the environment is the soil, and again, the factor is going to be the pH. So, you know, another way to look at think about environmental effects, right? You have genes to be tall, okay? However, you are not tall, you end up being short. And the reason you end up being short is because you're undernourished or you're malnourished, okay? So even though you have that genetic potential, you may not achieve or reach that genetic potential if the environmental factors are not there to support or influence that, okay?